lactose metabolism, three enzymes, beta-galactosidase, permease and trans-acetylase are very important in lactose metabolism. Beta-galactosidase breaks lactose into galactose and glucose. Permease permits lactose molecule inside the cell. Trans-acetylase transfers acetyl group from acetyl coenzyme A to galactose site. If lactose is present, these three enzymes are synthesized. If lactose is absent, these enzymes are not synthesized. Synthesis of these three enzymes are controlled by three structural genes Z, Y and A. Gene Z encodes beta-galactosidase, Y encodes permease, A encodes trans-acetylase. Three structural genes are regulated by repressor protein. If lactose is present, repressor protein is inactivated. If lactose is absent, repressor protein is activated. Repressor protein is produced by the action of regulatory gene I, inhibitor. Expression of gene Z, Y and A depends on whether operator is switched on or off. If operator is switched on, means if repressor protein is inactivated, it will not bind to operator region. RNA polymerase will move from promoter to structural genes over operator gene. Since operator is not blocked by repressor, RNA polymerase transcribe three structural genes into single mRNA. mRNA by translation produces three enzymes to metabolize lactose. But if lactose is absent, inducer is absent, operator is switched off, enzymes for lactose metabolism is not required. Let's see how. Regulatory gene I by transcription produce repressor mRNA. Repressor mRNA by translation produces repressor protein. Since there is no inducer, lactose, repressor protein binds to the operator region. Operator is switched off. Promoter region genes where RNA polymerase is bind cannot move to the structural genes because there is blockage, that is, operator region is binded by repressor protein. Due to this, structural genes are inactive. There is no synthesis of mRNA, so no synthesis of enzymes required for lactose metabolism because lactose is absent. So now let's see again. Inducer. Allolactose is present. Regulatory gene by transcription produce mRNA, which by translation produces repressor protein. Since inducer is present, it binds to repressor protein and inactivate repressor protein. Inactivated repressor protein cannot bind to the operator region. RNA polymerase moves from promoter to structural genes. Since operator is on, there is no repressor protein. RNA polymerase activate structural genes. Structural genes transcribe into mRNA, which translate and forms enzyme beta galactosidase, permease and transacetylase required for lactose metabolism. Pancreas Jacob and Jacques Monod in the year 1961 explained Operon in E. coli. So when lactose is present, operator is on, and when lactose is absent, operator is switched off.